Welcome back to part two of chapter one. So far, we've talked about some history and some historical players in microbiology. We're going to finish up with two more. So we're going to discuss the idea of Semmelweis, and we're going to talk about Joseph Lister for a minute. So in the 1840s, it was a very big issue for nosocomial infections. And nosocomial infections are hospital-acquired infections, meaning you picked up a bug in your stay. And what Semmelweis was able to hypothesize were that medical students were actually transferring these cadaver particles to delivery rooms, and moms that were in those delivery rooms were developing what's known as puerperal fever. Puerperal fever is based upon a bacterium that we know as Streptococcus. And so he started to notice this correlation between those that were working in the autopsies and working with cadavers, and then going into delivery rooms and this really high mortality rate. So what he decided to do is to make students, especially medical students in particular, wash their hands with chlorinated lime water. And this just drastically dropped those mortality rates. Within 1 year, it went from 18.3% to 1.3. So this is pretty amazing. However, doctors truly didn't believe that they were the cause. So there was not a lot of support based upon this method. So later, Semmelweis ended up developing depression. He was then committed to a mental institution, and ironically, he actually died from a streptococcus infection. So, moral of the story, make sure to wash your hands and try to prevent nosocomial infections. Another big person to make mention of is Joseph Lister. Joseph Lister was really involved in trying to prevent surgical wound infections, especially during the Civil War. If you think about the Civil War and think about if you had an open wound at that period of time, a surgeon didn't have proper cleaning techniques or what we know as sterilization techniques. They probably used a dirty rag. There may have been even contents left from the previous patient. So what Joseph Lister did is he introduced a system to really prevent any of those wound infections. And this is known as aseptic technique. These are techniques that are going to help to prevent contamination by any unwanted microorganisms. So the idea is to try to keep as little contamination or no contamination as possible. And he's stated to have saved more lives in the 19th century than all of the wars during that time had sacrificed. So big thanks to Joseph Lister. In fact, actually, as a thank you, Listerine is actually named off of Joseph Lister. No family relation, just kind of as a thank you. All right, next we're going to talk about microscopy. We're going to be doing a lot of microscope use in lab, so it's really important to understand the importance and basically how the microscopes are going to be used. So the first thing that we're going to do is we're going to talk about three factors that help to determine the quality of your image. First that we're going to talk about is magnification, so how to kind of amplify the picture or magnify the picture. We're going to talk about resolution, and then we're going to make mention of contrast as well. So when we think magnification, we're magnifying, like if you were to use a magnifying glass to make the image larger. And there's two lenses that we're going to use in what's known as a light microscope or a compound microscope. This is what we're going to use in lab. You're going to hear about the objective lens. That's going to be part of the nose piece. And what you'll see here is there's different objective lenses. Usually we start with something that'll say a 4x, then it goes up to a 10, a 40, and then 100. Those are the typical compound microscope objective lenses. And we see that in the nose piece. There's also going to be an objective lens, or I'm sorry, an ocular lens in the eyepiece. And this is going to be about 10x. So what ends up happening is we multiply the magnifications together to get the total magnification. So you would use the magnification of the objective lens and the magnification of an ocular lens. Let's give an example, for instance. So the ocular lens, or the eyepiece, is going to be 10x. Let's just say hypothetically we're starting on the most simplest objective lens, which would be a 4x. Our total magnification would be 40. If we moved on to the 10x and we had 10x for objective and 10x for ocular, our total magnification would be 100x. Now let's go to the highest magnification possible, which would be the 100x, the oil immersion. If you had that as your objective and you still had that 10x for the ocular lens or the eyepiece, your total magnification would now be 1000x, because you're multiplying the two together. So when we discuss total magnification, you have to multiply the objective lens by the ocular lens. All right, so this just kind of gives you that idea of how to see these pieces. And we're going to be utilizing some metric units as well for length to sort of understand this a piece. So this is just a brief table kind of helping with those metric units and sort of how we can utilize them and understand and actually convert them. All right, and this just kind of shows you the idea of how we can utilize that microscope and be able to actually see our image. What you'll notice with your specimen is using this microscope, the picture is actually inverted, it's reversed, and you'll see that the image itself is much larger than how it truly appears with the naked eye or the unaided eye. All right, we're gonna also talk about resolution. This is the ability to separate two objects that are really close together, so they are actually seen as two objects. They're not blended together in kind of like this mushy, murky piece. And the greater the resolution, we're going to see with those higher magnifications. So in the lab, we're going to be utilizing the oil immersion magnification. That's the 100x, and we'll actually use oil to help to really create this great contrast or this resolution between those two, so we can see them as two separate objects. And this just kind of shows you what happens without utilizing oil immersion, and then finally when we will use oil immersion at that 100x. And then finally, we talk about contrast. This is going to be the differences in intensity between two objects. So you can see the object and then you can see the background behind it. And a lot of times we will use staining to have that contrast. So usually microbes are going to appear colorless against a colorless background. So you're going to see like a very light colored microbe and a light background. So it's really hard to see. So the idea would be to use staining. And with the staining, it can either color the microbe and then you have a white background, or you can color the background and have a white microbe in order to see. So this just kind of shows you that idea. Figure A that you see would be if you didn't use any staining techniques. Again, it's really kind of difficult to see those microorganisms. You can see them, but you don't see them in great detail. Versus with that staining, it really allows you to see the background versus the microbe. And again, we can stain the microbe directly, or we can stain the background, just depending on the type of stain that we use. So when we discuss the idea of microscopy, there's a couple different types. What we will do in lab that's the most common, it's really easy to use, is a light microscope or a compound microscope. This is called a bright field. So what you'll notice is you have a very light background and you'll be able to see with staining those microbes that are going to appear darker. And this is really commonly used, and it's a really important tool for actually studying living organisms. And again, the highest magnification that we're going to see, highest total magnification, is 1000x. We get 100x from the objective lens, and then we're going to have 10x from that eyepiece or that ocular lens. You also can utilize an electron microscope, and this will magnify very highly, up to 100,000x for the total magnification. So this guy allows you to see lots of details. Usually you can see details on the cell surface. So for instance, if you were to do a Google search of the coronavirus, the reason that you can see the details of the spikes in that outer surface is it's utilizing an electron microscope. These are also very expensive. Usually they're $100,000 a piece. But this will be the guy that we use in lab. So again, this is your typical light microscope. And there's many parts on it that I would like you to know, not only for your lecture component, but also understanding for lab as well. So you should know the eyepiece, the ocular lens, know the objective lenses, know the stage where we actually put the specimen on, what the light source does, how it controls the light, the condenser as well that helps to focus some of that light. We're going to see what's known as an iris diaphragm that actually controls the amount of light that enters the objective lens. And then finally, our actual control for the intensity of light, our little knob that you're going to see. We're also going to talk about the coarse objective, or the coarse adjustment knob and the fine adjustment knob in a little bit more detail, but we will worry about that a little bit more later on. Okay, so when you see microscopy for the electron microscope, we're usually going to see TEM, which is going to be transmission electron microscope. And again, this is in really high resolution. You're going to be able to see internal structures. versus the scanning electron microscopes, again, also allow for very high resolution, but these are more useful for surface structures. So in this image here, what we'd be looking at is the TEM, that transmission electron microscope. And these guys here would allow you to see that scanning where we can see those cell surfaces. And when we talk about microscope techniques, we're going to utilize dyes and staining. And again, the whole reason why we dye or stain is to create that contrast. So again, we have a very clear background versus a very clear microbe that we're looking at. And it helps to create that resolution where they appear as two separate components or two separate images. So that way they're very visible when we look at those microorganisms. And usually microorganisms are transparent on a slide, so it's very important to create those stains. A couple stains that we're going to do are simple stains. So we could just put one type of stain on there, like crystal violet, or we could do methylene blue. We could do a safranin as well. But we can also do differential stains as well. And one of the stains that we'll be doing, or techniques of staining that we'll be doing, is gram staining, where we actually use two or more stains to provide contrast, and it allows us to see differences in bacteria. For instance, with a gram stain, we can either have the microorganisms, in particular bacteria, stained purple or pink based upon their cell wall. So we'll talk more about that in a couple chapters. And then we do have some special stains as well, where we can actually stain the cell structures, or again, we can even stain the background as well. So a simple stain, again, just allows you to actually see the cells. And again, it's based upon this like really bright background, and then you have this dark staining cell. So again, we can use methylene blue, crystal violet, safranin as well. And again, these are very useful as one stain that you're just placing on the microbe to be able to see the cell structure and arrangement. Differential stains, again, allow us to see differences in the type of microorganism, in particular bacteria. So the most common that you're going to see done, and we will be performing in lab, is a gram stain. This allows us to see differences between gram-positive and gram-negative bacteria. Gram-positive bacteria will stain a purple color, gram-negative will stain like a reddish pinkish color. There are some microbes that don't have the typical cell wall, like Mycobacterium. So we would do what's called an acid-fast stain, and we can see them either stain like a reddish or a blue color. And then finally, we can do an endospore stain as well to see if certain microorganisms, in particular, again, bacteria, house what are known as endospores. The endospores are going to help actually make the cells a little bit more resistant. It allows them to lie dormant for long periods of time. But it will stain a particular color. That spore will stain a particular color. So we can do those differential stains to see those differences. So again, with a gram stain, you would stain with two colors. We would use crystal violet, and then we would use safranin. The purple color will be because of the crystal violet. The pink will be because of that safranin. And it allows us to see the difference between gram-positive and gram-negative bacteria. Acid-fast stains, again, are used for mycobacterium, like, for instance, TB is Mycobacterium tuberculosis, and they don't take up the dyes and stains correctly for like a gram stain to be performed. And it has to do with this mycolic acid in their cell walls, and it kind of resists staining. So what we do is an acid-fast stain as well. And then finally, again, we can do those endospore stains. You can see the green structures inside of these bacteria, and that will allow us to see those endospores and what cells actually can form endospores by that malachite green. And then finally, we can also do what's called a capsule stain. So some microbes will have a capsule or like a kind of a coated protection over top of them. It's called a glycocalyx or capsule or a slime layer. And we can run stains to actually see that because the capsule can't hold the stain, and it will kind of highlight as almost like a halo around the bacterium. All right, and then finally, we can also do a flagella stain. So when we do a flagellum stain, what we are looking at is to see if they have a particular tail. These tails are used for motility, and they're known as flagella. Some microorganisms will have one, some will have one on one side or many on one side. Some can have them completely surrounding them. And so the flagella stain actually is used to stain the flagella, which normally, again, it's too thin to see without that additional staining added. All right, that's all I have for chapter one. I hope you enjoy these videos. Feel free to go back and watch them a few times. And I have incorporated some additional videos and some extra resources in your e-learning module. Thanks a lot and have a wonderful day.